Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the translation process. Okay, so here in this video I will try to describe about the translation process initiation. Okay, and generally you can here you will see the different terms or different factors that are involved with this initiation process, but here I only describe the process, but the factors which are involved in this process, these are already described in my previous video, so you just check out that one. Okay, so here the translation process is that the first one is the initiation. So what is the initiation? Initiation means that it involves a binding of the small ribosomal subunit to mRNA and then is joined with the large subunit. Okay. In case of ribosome, there are two subunits. One is the small, another is the large. So first one, this is the small subunit, ribosomal subunit that bind to the mRNA and then that joined with the large subunit. Okay. So it involves the reactions that precede the formation of the peptide bond between first two amino acids of the protein. Okay, so first the process, how the continuing, that the two ribosomal subunits that are involved here, they join with each other and then the involvement of the reaction that preceding the formation of the peptide bond, or you can say that the peptide bond formation is seen between the two amino acids. Okay, and then this process become continuing. Okay, so in case of Eubacteria, 30S, that is the small subunit, and the 50S, that is the large subunit, the two subunits are needed, and the two subunits associate at the ribosome binding site includes the upstream Shine-Dalgarno sequence. This, what is this Shine-Dalgarno sequence? This is nothing but the polypeptide sequence that is starting with the 5 prime and it is ending at the 3 prime. And in the middle there are the nitrogenous bases are there. That is this is the example AGGU. Okay, 5 prime AGGU 3 prime. So this type of sequence, the small short polypeptide sequence, this is known as the Shine-Dalgarno sequence. So what happening in here, the two subunits that bind with each other, where they bind? They bind with the short polypeptide chain of the upstream. You all know that there are two stream, upstream and the downstream. 5 prime to 3 prime, that is called the upstream, and downstream is the 3 prime to 5 prime. So always they bind with the upstream. And here the process is in eukaryotes. I have written stepwise, so you can easily understand that the process. So in first case, or in the first step, the small subunit recognize the methylated cap that marks 5 prime end. Okay, so methylated cap means that remain. Cap means the first portion that is started with the polypeptide, the started codon, you can say, at the polypeptide chain. First codon is called the cap and the last one, this is called the tail. So the methylated cap that marks 5 prime end of the mRNA. Okay, and then the scan then the scan along the sequence until it locates the initiation codon. So what is the function of this? Just bind with the methylated cap, which one? The small subunit, and the target is what? The target of the small subunit is to identify the initiation codon. Okay, if the initiation codon is there, that means the small ribosome fixedly bind there. Okay, so the second one is that the scanning process is done where the initiation codon is there. The third is that they are joined by the large subunit, and if the initiation codon is totally identified or this is recognized the small subunit, then the large subunits bind also there. But this process is is done by the enzyme that is called the helicases. Okay, and here obviously the hydrolysis of ATP is occurring. Okay, and the next step is that what? The selection of the AUG is facilitated by the specific nucleotide, and this AUG is this AUG is the initiation codon. Okay, so selection of AUG is done by some sequence. This is the sequence known as the Kozak sequence. And the next step is that once the small ribosomal subunit with its bound MET tRNA, MET means what? MET means methionine. tRNA is correctly positioned at the start codon, union with the 60S means large ribosomal subunit that completes the formation of 80S subunits, 80S ribosomal subunits. That means here the total ribosomal subunit formation is completed. The 80S is the complete ribosomal subunit, where the small subunit is the 30S, I think 30S. Okay. 1 second. 30S, yes. And okay, I'll just check the small first. I'll just check the small subunit is 30S or not. Okay. Okay, so this is the 40S subunit. Okay, no 30S. This is the 40S subunits. Here in the eukaryotes, this is the 40S subunit. The small subunit, that is the 40S. So what I am saying that this 80S ribosomal subunit that is completed here. Okay, in eukaryotes, and here another thing is that written in Eubacteria. In Eubacteria, ribosome bind directly to the start codon in the interior of an mRNA molecule to initiate the protein synthesis. Okay, in case of Eubacteria, and in eukaryotes, in Eubacteria, the ribosome directly bind to the start codon and then initiate the protein synthesis. In Eubacteria, an alternative means of initiation process takes place, where a small subunit, that is the 40S, that associates directly with an internal specialized sequence. Internal specialized sequence is what? This is the internal ribosomal entry site. This is the internal ribosomal entry site, or short form is that IRS. This is the alternative process, and it is not exactly happen to the eukaryotes always. This process is happen generally in the eukaryotes. Okay, so next part is that initiating amino acid and the initiating codon. In E. coli and other Eubacteria, the first amino acid the first amino acid of the newly formed polypeptide chain, that is the N-formyl methionine, and AUG, GUG and UUG that serve as the initiation codon in the E. coli. Okay, so in the E. coli and other Eubacteria, the first amino acid is the N-formyl methionine, AUG and GUG and UUG that serve as the initiation codon in the E. coli. Okay, so this is just first thing of the initiation process is explained here, and it is continuing, and I have to just wrap this one, and then I will discuss the next part of this initiation factor. Okay. Okay, so here the initiation process is briefly described in the prokaryotic, obviously. So in Eubacteria, mRNA binds to the 30S subunit, and this is the rough diagram. Okay, first mRNA binds to the 30S subunit, then binding is stimulated by IF3, but here both initiation factors are involved, IF3 and the IF1. Next step, the initiation codon lies within the P site. You can see that initiation codon that lies within the P site. and the tRNA enters into the P site, and next step the tRNA, that is the tRNA, enters the P site. Okay, and IF2 places that tRNA at the P site, and IF2 is involved here to place the tRNA at the P site. IF2 is very important here, that helps to place the tRNA in the P site. Okay, and when the large subunit joins the complex, the P site tRNA converts into the P and the A site. Sorry. Okay, so here A site tRNA and complementary to the secondary second codon. And here what happened, that you can see that the P site now is completed, because the small subunit, the large subunit also added here, and the complex is totally, ribosomal complex is totally formed here. Okay, and here the P site that tRNA, that converts into the P and A site, and this A site tRNA that is complementary to the second codon. That means in the A site there the second codon is trying to add or try to join there. Okay, so these are the steps of the initiation, and it is happening in the Eubacteria. There are some differences between the eukaryotic cell obviously, and we will learn about what are the differences between the prokaryotes and the eukaryotes in my next video. Okay, because this will become very large if I try to elaborate the eukaryotic transcription initiation process, so it will be large. Okay, so I will try to explain the eukaryotic initiation process, or the differences between the eukaryotic and the prokaryotic initiation process in my next video. Okay, so here one thing is also written, that AUG, GUG and UUG, these are the start codon, you can say, and also you can say that the initiation codon. Okay, any of one is recognized by the N-formyl med tRNA or methionine tRNA, and N-formyl methionine appears as the first amino acid in the chain. Okay, after identifying any one of the start codon, that methionine, that is the N-formyl methionine, appears as the first amino acid to bind with the any of the first start codon. And here the AUG codon is not what I am saying, that it is not included with the initiation factor. It is just explained here that if AUG is added as the initiation factor, then what we call them, or how we read as AUG, or if GUG is added as the initiation factor, then what we call them. Here, if AUG codon is used for the initiation, then read as the formyl methionine, and when it is used in the coding region, it represents the methionine. Clear? But in case of GUG, that serves as the initiation codon, it is read as the formyl methionine, NINE. But when it present in the coding region, it codes valine. Okay, valine and methionine, these are all the essential amino acids. Okay, and here I try to explain that initiating tRNA, and the structure of tRNA already I have explained in my previous video. You just search and check out that video. Okay, here just I will try to explain that initiation factor. As initiation factor, what is the role of the tRNA? The both prokaryotes and the eukaryotes, the tRNA carries the formilated methionine residue, and that initiates the protein synthesis. Okay, so tRNA that carrying the methionine residue, without methionine this protein synthesis process is not started, and it recognizes the AUG or GUG. Already I have explained there, which and and the acceptor stem of this tRNA that helps to bind with the IF2 or the initiation factor 2, and the base pairs of the tRNA that require to allow the tRNA to insert the P site. Okay. So this is the actual explanation, or you can say the just brief explanation of the initiation process of the translation in the eubacteria or the prokaryotes. Okay. So in the next video I will try to explain about the eukaryotes. If this video is liked by you, or you can understand properly the initiation process, so please you share this video, and you are new one in my channel, so please subscribe for the growth of this channel, because without your support I cannot continue this channel. Okay. And another thing also is that you can comment in the comment box how this video is important to you, or how can you understand this. So if I get your comments, then I will try to explain the any topic in the easier way. It is typical, that means I want to say that if this is explained clearly or not. If it is explained clearly, then you can mention, or it is not, then also you can mention. So I will try to explain properly towards you. Okay. So thank you.